I'm Konrad. As I was introduced, I'm from the Molecular Mechanism cluster up in Edinburgh, which is one of the clusters funded by the Human Functional Genomics Initiative. Before I tell you about our project, I briefly should mention some conflicts of interest, in particular here Multiscale Labs, which is actually a spin-out kind of out of the Functional Genomics Initiative partly, and which is taking some of the aspects of ideas underlying the MMC, which are perhaps more speculative or harder to scale in academia, and trying to see how they can be scaled in industry. But let me talk about the MMC. So what's the purpose of the MMC? At its core, it's a very simple project. I haven't kept count, but I'm not going to be the first one today telling you that we know of a lot of genetic associations with disease. And for many of them, we do not know how to translate them into therapeutics. And the kind of fundamental reason really is that we do not understand the kind of cascade of molecular events that mediate these whole phenotype events. So the purpose of the MMC is to essentially generate data which should help to close some of this gap. And we do this in a disease-agnostic manner. So we're not trying to explain the associations for a specific disease. We're aiming to generate kind of broad data that has enough breadth to act as a substrate for others to either explain the associations they find or to be used as training data for AI models to extrapolate beyond the observations in the data that we generate. All right. Oh, sorry. Goodness. So what this kind of means in practice is that we are generating molecular readouts at a sufficient scale to essentially allow mapping of molecular QTLs, so associations between genotypes and molecular traits. And scale here is really twofold. There's a kind of mundane scale of we just need enough samples to do mol QTL mapping. But the other scale that also was repeatedly mentioned today is that molecular events happen in a context. So a molecular event happens in a specific cell type. It happens in a specific cell state. Expression of inflammatory pathways happens in response to a stimulus, and to observe the effect of mol QTLs on these pathways, we really need to observe the expression in this context. So with these two kind of scales in mind, the MMC focuses on assaying RNA expression at single cell or rather single nucleus resolution, this being currently the kind of most cost-effective approach of assaying the molecular state of a cell. And in addition, we don't just look at any cells, we look at cells in disease-relevant tissues. This was originally 4 tissues, so brain, lung, skin, and PBMCs. But PBMCs are relatively easily accessible to anyone. So we are focusing on the other 3 now, because it's not just any tissue. As I said, state is important. So rather than looking at cell lines or looking at organoids or looking at post-mortem tissue, we kind of argue that really the state you're interested in is when the tissue is functioning within a living human, either healthy or ill, but still very much alive. Obviously, we cannot assay tissue in situ. Most people do not want to give part of their brain away. But the aim of the MMC, what we strive to do, is to kind of get as close as possible to that at scale. So what this means in practice is that we work with the NHS, we work with surgeons, research nurses, pathologists to obtain excess tissue from operations. So for example, if you're having a tumor removed, the surgeon can't just move the rest of the brain out of the way. A bit of nominally healthy brain has to be removed to gain access to the tumor, and we get this healthy access tissue, and then perform single nuclei sequencing on it. In addition, we perturb the tissue with various kind of disease-relevant stimuli to further broaden the kind of space of cell states, which hopefully are disease-relevant that we observe. I hope everyone can appreciate that the process of getting tissue that's being taken out of somebody who's undergoing an operation is not entirely straightforward. We have kind of spent a substantial amount of time building up a set of protocols and procedures to make this work efficiently and relatively smoothly now. We're recruiting at a rather good rate, I would say. We'll easily hit the targets that we need for eQTL mapping. And in fact, for some of the tissues, we probably have an oversupply of tissue, and that allows us to generate some additional data, which I'll talk about later a bit. Originally, we started this just in Edinburgh, but this has and is kind of continuing to grow into a more nationwide network for fresh tissue supply. And we would very much invite anyone who is either interested in accessing such tissue or would like to help setting up access where they are to get in touch with us. We think that a network like that would be a kind of great legacy from the whole initiative even, because this tissue is, you know, it's excellent. What do we do with the tissue once we have it? I mentioned that we are interested in two things, mapping single-cell eQTLs, but also doing perturbations. Each of these requires samples. If you want to do both, you need a lot of samples. Therefore, we kind of decided on a two-phase approach where we first do more perturbations with fewer people, kind of assaying the landscapes of contexts, and then doing just a few perturbations on many people to get enough data for eQTL mapping, and then integrating the two together to get a broad overview. We very much see this project as a kind of platform, and as such, all data will become available as soon as practically possible, really. In particular, as was already mentioned earlier, we have a collaboration with Open Targets to put any QTLs that we find directly accessible in a kind of broad, well, the broadest available, I guess, translational context out there. Right. An unfortunate aspect of a project like this is that it's very much of the kind of good things come to those who wait nature. We just can't map eQTLs with a few samples. So where we are at the moment is kind of finishing the pilot phase and starting the delivery phase, or rather have already started the delivery phase. So we're just pushing the last samples through the sequencing in the pilot phase. So we don't have all the data from there yet. And we are queuing samples for the delivery phase. With that in mind, I'll just briefly talk through some of the data we have, keeping in mind that it's not a complete data set yet. So we've so far sequenced about 5 million nuclei across the 4 tissues. We have, in terms of sequence, different numbers of conditions for the different tissues, but about 5 to 10 perturbations, with some of the perturbations also having time course data. And I won't go into too much detail about the analytical approach we're taking, but as an outline, what we're doing is we're using deep learning models to integrate the data across donors and perturbations. That's what's shown kind of here for skin. And these models do not just allow us to integrate the data, they also allow us to learn the effect of the perturbations so that we can consider counterfactuals. That is, we can take a cell which was observed under perturbation A and then apply a simulated perturbation B to it, allowing us to compare the effect of different perturbations in the same cellular context, at least up to the level that the model has learned it. And if we look at the effect of the different perturbations, Here again for skin. So this is once you, the previous slide was the data kind of integrated, and this is once you apply the perturbations to these cells. Then you see that most of the perturbations have kind of really a within cell cluster effect, but I would like to draw your attention to one particular perturbation over here, which is this uncultured condition, which is essentially the freshest tissue we have, and which is very much a thing on its own. It is in not just in skin, in the other tissues as well, the most different condition that we see. You can kind of summarize that by considering the similarity of the effect of two perturbations across all cells. And we kind of see expected natural groupings, but we also see that, again, the fresh tissue is very much different to everyone else. And we're kind of working towards, right now, building a kind of progression timeline from uncultured to post-equilibrium to the standard culture conditions and hypoxia conditions to get an overall picture of what are we losing or what's changing over time as the tissue ages. Well, ages in the sense of ages from harvest. In fact, for that purpose, we, I think just this week, perhaps collected additional tissues, which are literally minutes after being removed from the living context. And these will be sequenced once they're processed, and will give a kind of starting point, hopefully anchoring the entire thing. For the other kind of disease-relevant perturbations, we are also seeing promising results. Different perturbations show cell type-specific effects, largely in the cells you would hope them to have an effect. We are kind of waiting for the last sequencing for the pilot to happen and working our way through the data, so watch this space, hopefully. But also, if you think that there are questions that this kind of data could help you answer, bearing in mind that we can't really do much genetics yet because these are small n's. Do get in touch early. We are very open to work with others on getting the most out of this data. And finally, as I mentioned, we have probably a bit more tissue than we need, allowing us to do other things to kind of broaden the scope and put even more context on the data that we have. So we're working with Oxford Nanopore on two things. One is to get matched long-read single-cell sequencing for some of the samples we have to provide kind of scaffold of isoforms that are in there that we can use to get more out of the short-read data. And we are going to try some direct RNA-seq on the fresh tissue to have a look at the RNA modifications in there. And if you need a job and these kind of things are of interest to you, we are actively looking for people to join on this project and on other projects in the Pandemic Science Hub. And with that, I would like to thank in particular all the patients and all the NHS staff who make this even possible, and then also all the collaborators who work on the specific tissues and have shared their expertise in how to handle those individual tissues. Thank you very much.